Good morning, everyone. My name is Technique and I am the student of Christianity College. I'll be presenting a seminar on microarray technology guided by Dr. Manigandan sir. Introduction. DNA microarray is a collection of microchips or DNA spots attached to a solid surface. They are used to study mutations. They are also called as biochip or DNA chip, used by researchers to obtain faster results for gene expression analysis. They can be like, if you have a sequence of DNA which has to be analyzed more faster in a more efficient way, your results can be obtained with the help of biochip. The principle behind the use of DNA microarray technology is that for microarray to check the transcriptome of the cell. It is used to check the transcriptome of the cell. What is the transcriptome of the cell? It refers to the total mRNA content of the cell at a given time. The principle followed is DNA hybridization, where your mRNA is used as a surrogate marker, in which your mRNA serves as a surrogate to form a cDNA, after which you follow the process of DNA hybridization. Two fluorescent probes such as cyanin 3 and cyanin 5, that is CY3 and CY5, can be used. The unhybridized sequences are removed by multiple washing. Then analyzer is used to detect the expression by checking the intensity of light. Preparation of microarray slides. What are the two steps of preparing micro? First is masking. First is blocking. Second is masking. Blocking refers to is performed to prevent non-specific binding between extracted nucleic acid and labeled probe. A blocking agent is added to the slide to seal the wells. Blocks can be removed by laser treatment. Masking is also done to avoid non-specific binding, in which you create gaps at alternate sites. Like the site where you need your cDNA to get fixed is you create a gap, then you mask other cells, and then you add your probe. Then the nucleotides act as a probe to which the sample DNA attaches. Instead, it can also be used as single strand, which will be complementary to the extracted DNA sample. Both of the probe and sequence will be known, which will be helpful for screening. Techniques like how you can introduce a spot on your microarray can be by pin technology, pin ring technology, quills or slip pins, jetting technology. In pin technology, it is the oldest technique in which you use a injection type thing, where the injection sucks your spotting material and transfer it to your required region. Pin ring technology is more efficient because when it comes to pin technology, your sample may get evaporated. As it is a small amount of sample, it might get evaporated or it might not be stable over that pin. So when it comes to pin ring, the other external factors affecting that material will not be working more anymore. Quills and slip pins, a solid pin, like it sucks, and just like your pressure, you apply a pressure and it sucks your material, and then you place the drop over the slide. Then is your jetting technology, which works like an inkjet printer. So this is your pin technology, pin ring technology, quills, slip pin technology, jetting technology. Then your protocol for performing microarray technology is that collection of, you first extract your mRNA can be extracted by your um, your technique of gel electrophoresis, like bead electrophoresis, you can perform that thing. And then you get your mRNA. Once you get your mRNA, you convert that into cDNA using reverse transcriptase. Once your mRNA is converted to the your reverse transcriptase, you um, add your total your case sample as well as your control sample, and that means you have your control sample which is from healthy cells, and you have your case sample, the test sample. Then you add both of the samples to the microarray chip. The both of the cDNA obtained from that samples are added to the cDNA chip, and then you do your screening, which is the study of your fluorescent probe, and then you obtain your results. So this is a flow chart about explaining about how your cDNA works. Your DNA microarray works. This is your observation result. So when it comes to red color indicates the greater binding of the test sample and showing an upregulation of gene. Yellow color indicates equal binding between the sample and showing a constant regulation. Black indicates no hybridization. Green indicates laser binding, down regulation of gene. Expression levels, like how the formula for calculating expression levels is, that is expression levels is equal to test sample intensity upon control sample intensity. Then your types of microarray are glass DNA microarray and high density oligonucleotide microarray. Microarray classification of based on function, they can be used for expression analysis to how extent your gene is expressed. Comparison is between control and diseased tissue. So like if it's a genetic disorder, you can coming to know that what type of genes are included in the expression of your following protein, which leads to disease. Mutation analysis, that is SNP. You check your single nucleotide polymorphism using your genomic DNA. The analysis is performed to study the genes might differ from each other by less as single nucleotide base. Then your comparative genomic hybridization for check like increase or decrease on any chromosomal number of fragments. So there are two types based on experimental factors, that are oligonucleotide and spotted microarray. Typical probe is 18 to 30 mers, whereas your spotted DNA, the probe can be of 500 to 1000 base pair cDNA. Spot density is higher for oligonucleotide. Hybridization is higher for oligonucleotide. But when it comes to expression levels, your oligonucleotide microarray is single channel, whereas spotted microarray is dual channel. But advantages are more for oligonucleotide as it is single channel, no mixing up of the sample or no uh, variants are less. Then but when it comes to spotted are much cheaper than oligonucleotide. You can use your both control and the case sample on the same slide. But as I said, there are uh, problems related to dual channel based microarray when it comes to higher efficiency and verification. Applications, they are used to detect cancers. They can be used as tissue microarray, proteome microarray, drug action. These are my references. Thank you.